Okay, this session has to do with the structure and function of macromolecules. Macro means big, it means big molecules. And the biomolecules that make up life are macromolecules. So we're going to look at some of those and their purposes and how they're formed and how they're broken down. Some definitions. If you remember, definitions like organic, that means that they're molecules or compounds which contain carbon. Molecules of life are organic. Inorganic means that they are molecules or compounds which do not contain carbon. Inorganic, so they do not contain carbon. Now, a macromolecule is a large molecule. It means large. These are large. I mean, they're pretty sizable. The biomolecules are macromolecules, and they're different types of biomolecules. We have 4 groups of biomolecules. We have carbohydrates, proteins, nucleic acids, and lipids. Those are the biomolecules, molecules of life. Those are consistently found in life as we know it. Now, the building blocks are called monomers. That means one. Mono is one, and mer means unit. These are the building blocks for all of the biomolecules. When you put building blocks together, you make what's called polymers, which means many units. Now, poly usually applies to 3 or more. 3 or more monomers makes a polymer. Okay, and these biomolecules and macromolecules, they are polymers of these building blocks, the monomers. So the monomers are the building blocks, which we'll talk about this, and they're put together to make polymers, which are the large molecules, the biomolecules. Now, the first chemical method is called condensation. Now, condensation is also, another term is dehydration synthesis. If you've ever heard that word, it's the same thing, but I'll use condensation here. A type of reaction in which a hydrogen is removed from one molecule while an hydroxyl is removed from another. The result, or as a result, both molecules bond together by way of a covalent bond. Now, when the hydrogen is taken off of one and a hydroxyl is taken off of the other, what do you think that can be made? They can be put together to make something. Water. And water is condensation. You know, when it rains, that's condensation. When you take a can of Coke outside and it gets moisture on the outside, that's condensation. You have a broken windowsill, that's called condensation in your window. So where the hydrogen and hydroxyl were taken off, those are used to make water. And then where the hydrogen was taken off of one and where the hydroxyl was taken off of the other, a bond is formed between those 2 molecules, like it says. As a result, both molecules bond together by way of a covalent bond. So let's look on the next slide. Here it is. Let's read it again. Condensation, a type of reaction in which a hydrogen is removed from one. Look down here at the fructose. See that's the little hydrogen circled? Okay, molecule, and while a hydroxyl is removed from the other. Look down at that glucose. Hydroxyl is taken off. Those 2 together are going to make water, H2O. As a result, both molecules bond together by way of a covalent bond. Where the hydrogen was taken off of the fructose and hydroxyl off of the glucose, now they're bound together by that oxygen that was still hanging there. And it makes a disaccharide, a double sugar called sucrose. These 2 are simple sugars. They're called monosaccharides. You put 2 monos together and it makes a disaccharide called sucrose. So condensation is where a hydroxyl is taken off of one, a hydrogen off of the other. That makes water, that's the condensation, and then a bond is formed between that oxygen that's hanging there by itself and the carbon that the hydroxyl was taken off of. And there's your bond. Hydrolysis is just the opposite of condensation. Hydro means water and lysis means to destroy. This process is destroying a bond by putting water back in. Remember, condensation took it out. Hydrolysis is going to put it back in. So it's the opposite of condensation. Occurs during the digestion of complex molecules. So when you eat materials, you ingest food, and it goes into your stomach and your digestive system. Hydrolysis is taking place. You're taking in large biomolecules, large carbohydrates, proteins, nucleic acids that are in the cells, the nucleus of the cells, like in corn or beans or something like that, or even meat, and lipids. And through a process called hydrolysis, you're breaking the bonds between the building blocks that built it. The hydrogen and hydroxyl, which were removed through condensation, are added back to the molecule, thus breaking the bond that links individual monomers. So you're going to add the water back in. So that sucrose, which is a disaccharide, is going to be broken down by hydrolysis by adding the water back in into glucose and fructose. So look on your next slide. On the left, here's our sucrose, our disaccharide, and you see that there's that oxygen in the middle that's tying the two together. Well, enzymes, which we're not even concerned with those pathways, come in and they will break that bond between that oxygen and the molecule on the left. Another pathway will break that water down, which you can see on the right-hand side. Water is broken down into hydrogen and hydroxyl, and the hydroxyl is added to the glucose, and the hydrogen is added to the fructose, like they were initially. So the bond was broken. That disaccharide bond, where the oxygen is broken by adding water back in. And now you end up with two monosaccharides again. So you went from a disaccharide, double sugar, to two monosaccharides, single sugars. So hydrolysis is breaking the molecule down. Here's another view, just showing you if you like to see one view versus another. This is condensation. This top one, you see the short polymer. It's got 3 units to it, and a 4th unit is going to be added. Well, in condensation, the hydrogen is removed from one, a hydroxyl from the other. There's your water for condensation. And you have a bond that's going to form between the chain and the new monomer that was just brought in. And so now you have a larger, longer polymer. Now it's 4. So it's building. But hydrolysis, you know, is just the reverse. So you have this long chain, and it's going to be broken. So the bond's broken between unit 3 and 4, and the water molecule is added back in. So the bond's broken, hydrogen is added to one molecule, hydroxyl to the other, and you see that the chain is shorter now. So it's hydrolysis of a polymer is occurring there. So the same process that was shown previously, just a different format. This is shown with the little balls here instead of molecules. Here's just showing some neutral fats here. Neutral fats are fats that are around your body, like, you know, behind your eyes, around your heart, around your kidneys and your belly, your butt, your thighs. That's all neutral fat, just body fat. And fats are made up of 2 things. Glycerol, which is the 3-carbon alcohol. See those hydroxyls? Glycerol. And fatty acids. Fatty acids are long chains of carbon, and there's a carboxyl group on one end. See, there's your C-double bond OOH, a carboxyl group. Now, this is condensation that's occurring here to build this fat, because fats are made up of 2 things, glycerol and fatty acids. Those are the building blocks for fats, glycerol and fatty acids. Well, a hydrogen is taken off of one. There it is, circled in blue. A hydroxyl off of the other, off of the carboxyl group. That makes the water, and a bond is going to be formed between that oxygen and the carbon, which will be shown on the next slide. There it is. See, it's called an ester bond or ester linkage. So when you added 1 to that glycerol, that's a monoglyceride. If you added a second one, that's a diglyceride. And you add 3, that's your triglyceride. They call it triglycerol. Same thing, triglyceride. Got 3 fatty acids joined to a glycerol molecule. And that's a building process, so it's condensation. Carbohydrates. Okay, carbo says carbon, hydrates, hydroxyl, OH, and there it is, carbon, hydrogen, and oxygen. The ratio of hydrogens to oxygen is 2 to 1, the same as water. Remember I told you that glucose was C6H12O6, H12O6, that's 2 to 1. Now some varieties of carbohydrates. There are simple sugars here, which are called, they're the monomers, or the building blocks, and they're called monosaccharides. Simple sugars, one sugar, monosaccharides. And then we have disaccharides, oligo, and poly, and we're going to look at all of these on the next few slides. So here is an example of, through condensation, two monosaccharides, glucose and glucose, are put together by condensation. Hydrogen is taken off of one, a hydroxyl off of the other, and there makes your metabolic water. And you made a disaccharide called maltose. So maltose is made up of 2 glucose units. And maltose. Down here, you have glucose and fructose. Those are 2 different monosaccharides. But still, you have a hydrogen taken off of one, a hydroxyl off of the other, and there's your water. And it makes a disaccharide called sucrose. Okay, made of 2 sugars, disaccharide. Now, they're both disaccharides, maltose and sucrose, but they taste different. They're arranged differently, made of 2 different combinations of monosaccharides. One's made of 2 glucoses, one's made of a glucose and a fructose. So they're going to be, these disaccharides are structurally different. Sucrose would be like if you go to McDonald's and you get a shake, or you go to Wendy's and get a shake. That's what that tastes like. But if you go to Brahms and you ask for a malt, they use a different sugar. And you'll notice that maltose tastes different. A malt tastes different. It's different sugar, different sweetness. So this is basically showing monosaccharides, simple sugars. So it serves as a source of energy and as building blocks for more complex carbohydrates and as carbon chains for other biomolecules. So that's what they are. They're the building blocks. The monosaccharides are the building blocks for more complex carbohydrates. Now here's some showing you that not all monosaccharides are the same. See, these are called triose sugars. They have 3 carbons. Pentose, they have 4. Hexose, they have 6. Okay, so those are aldoses because they have the aldehydes, keto bond OH. Well, the ketoses is a 3-carbon sugar, and it's a ketone. See, it's got a keto bond O. Ribulose, like for RNA, ribulose, and then fructose. So all these are monosaccharides, 3, 5, and 6 carbons long. Disaccharides, food storage in many plants. Example, sucrose. This is what makes like oranges and apples have their sweet taste, pears, the sweet taste, even bananas. They're called disaccharides. And like we saw before, here are the disaccharides here, maltose and sucrose, made of 2 monosaccharides. Glucose and glucose makes the maltose, the disaccharides. The monosaccharides, glucose and fructose, make the disaccharide sucrose. Oligosaccharides. Oligo means few. So this has a few branches. It's a chain of monosaccharides, but they have a few branches involved in cell identity and other cell-to-cell interactions of the membrane. On the surface of your cell, they give you your cell identity for things like your blood type. They give you your cell identity for things like organ transplants. Different people have different oligosaccharides that are involved in cell identity, and you have to find somebody that has enough that are similar to yours in order for an organ transplant to even be achievable. Polysaccharides, it means many sugars. These are long chains like starches. It says food storage compounds, starch in plants and glycogen in animals like potato starch, corn starch, Those are long chains. And glycogens are polymers of glucose. So it is a polysaccharide. Or structural compounds, like in cellulose in plants, they have cell walls made of cellulose, which we cannot derive benefit from. That's the roughage in our diet. And chitin in insects and fungi. Chitin makes insects have hard shells. And insects have that, and fungi have it too. It's just that you don't, it's so thin you really can't feel it. But you know, insects are crunchy. That's their shell. So here is amylose. That's a starch, a long chain. Amylopectin. This would be like a oligosaccharide. It has a few branches on it. And here is glycogen. Ooh, a big polymer of glucose. Lots and lots of glucose molecules there. It's a quick energy molecule. Like it says here, glycogen is a storage polysaccharide in animals. Glycogen is stored mainly in the liver and muscle cells. Hydrolysis of glycogen in these cells releases glucose when the demand for sugar increases, because glucose is the main molecule that your cells prefer to break down for energy to make ATP. And that energy is used for things like muscle movement, you know, if you're being threatened. Here is starch versus cellulose. Okay, the top polymer is starch, and the bottom one is cellulose. Now you look at them and you say, well, gosh, those are just chains of glucose going across there. What's the difference? Well, how come one's called they're all 1,4 linkage, and let me have a little pointer here. There's the oxygen on this hexane molecule here. It's got 6 carbons actually, but the one's in the air. You start moving, counting clockwise. So that's carbon number 1 at that point, carbon number 2 there, carbon number 3, carbon number 4, carbon number 5, and then carbon 6 is in the air. So you count clockwise. Carbon 1, okay, there's a bond there. Carbon 1, carbon 2, carbon 3, carbon 4. That's where you get the 1,4 linkage. 1,4 linkage. Well, they both have a 1,4 linkage. So if they both have a 1,4 linkage of glucose, what's up? Well, one's called alpha and one's called beta. And if you look at the arrangement, look at how these all have the CH2OH on the top, and these are alternating. When they alternate, it forms a different 1,4 bond angle. We have the enzymes that will attack this bond angle. Remember, this is all three-dimensional, guys. It'll attack that bond angle. It will not attack this bond angle. This 1,4 is differently shaped than this one. The molecules are arranged differently. So our enzymes will break starch down to glucose for energy, or it can be stored as fat. But cellulose, we can't digest that. We don't have the enzymes for that. So that's going to be the roughage in your diet. When you eat vegetables and stuff, that comes out. You don't digest those. So down here at the reading, the polysaccharide cellulose is a major component in the tough wall of plant cells. Like starch, cellulose is a polymer of glucose, but the glycosidic linkages differ. See, they're different. That 1,4 is different than that 1,4. The difference is based on the 2 ring forms for glucose, alpha and beta. They're not put together the exact same way. And so that bond angle, even though it's a 1,4, there's enough difference there that our enzymes will work on one and not work on the other. Lipids, these are fats composed of carbon, hydrogen, and oxygen. Hey, that's just like carbohydrates. Oh, with a higher ratio of hydrogens to oxygen than in carbohydrates. So it's not the 2-to-1 ratio anymore, not 2-to-1 anymore. Lipids are insoluble in water. Well, you knew that. If you pour oil on water, it floats, right? You've seen that before. Now, there are different types or different varieties of lipids. The first one's called neutral fats. I told you about that one earlier. Involved in food storage, insulation, and shock absorption. This is the fat around your body that you normally just call fat. When you want to lose weight, you're losing neutral fats. It does afford some shock absorption, like behind your eyes, around your kidneys. It does afford a barrier there for shock absorption under your skin a little bit. and in food storage because you can get rid of it. When you eat too many carbs, they get stored as lipids. That's why you gain weight, and you got to get rid of it, and it's harder to break them down. There's a lot of calories in lipids. Phospholipids, now that's the one that forms your cell membranes. Primary structural component of cell membranes. We're going to see that it's a phospholipid bilayer. There's two layers of phospholipid molecules that form cell membranes. We'll look at that a little bit later. Some more, we have waxes. Okay, waterproofing and pliability of tissue. Now we produce a wax, it's called earwax, and it's called cerumen. So we do have some lipids that are produced by cells in our ear canals for waterproofing and pliability of the tissue, and they prevent bacterial infection too. Carotenoids, these are in plants, yellow to orange plant pigments. That's why they call them carotenoids, because most of the carrots that we eat nowadays are orange. They're not, all carrots are not orange, but the ones that are most popular are orange. Often involved with photosynthesis. We will see two in particular that carotene and xanthophyll that fall into the carotenoid grouping. That's a group of yellow and orange plant pigments. Then we have steroids, another type of lipid, involved with membrane structure and the regulation of metabolic activity. So when you talk about steroids, like testosterone, estrogen, that kind of stuff, those are steroids, and we'll look at those a little bit later. Here's our neutral fats. Remember, you are going to put together glycerol and fatty acids. Those are the building blocks. Glycerol and fatty acid, the building blocks of lipids, and they're put together by condensation. There it is, formation of a molecule of water. That'll make a monoglyceride. This is showing a triglyceride. It's happened 3 times, so 3 molecules of water were produced to make this triglyceride. How many molecules of water do you think it would take to break that down? Should be 3. You got to add them back in. Break that bond by adding the water back in, that's 1. Break that one, that's 2, and break that one. It'd take 3 molecules of water to break that down to glycerol and fatty acids. Types of lipids. Okay. Well, the first one over here is saturated fat. Now, saturated fat has only single bonds between the carbons, so it's a long chain of carbons that are maximally bonded to hydrogens. So it's a long chain, so each carbon has a bond to another carbon, but the 2 open ones above and below the chain are filled with hydrogens. So it says, and this is their example right here, long chain, there's all the hydrogens on the outside. Straight chains lay parallel to each other, which makes them more viscous at room temperature. That means it makes them thicker at room temperature, like lard. Or if you've cooked something in the skillet after you're through making it, and you've already had your supper, and you go back to clean up, and it's solid, fats in there. Those are saturated fats. Those are not good for you. Unsaturated fats, 1 or more double bonds in the carbon chain, and when that happens, it bends. There's a double bond there, so they're not maximally bonded to hydrogens anymore at that one point, and the chain bends. Double bonds cause bent chains. They tend to be liquid at room temperatures, like vegetable oils that you would use for, cooking things. You can use olive oil, vegetable oil. Those are all liquid. Those are all unsaturated fats. This is a slide that just shows you. Here's a saturated fat right here. See, there's a long chain of carbons, and they're maximally bonded to hydrogens. And here's your acetyl bond OOH on the end, the acid, fatty acid. Well, here's an unsaturated one here. And see that double bond there that's circled? It makes it bend. Makes a chain. Here's another one right here. It makes it bend again. So this is definitely unsaturated. This would be more of like a polyunsaturated. The trans is found in some margarine. Here's the double bond, but you see when there's a double bond, it's at a different angle than it is up here on the unsaturated. It makes a bond angle. It makes a chain bend between those carbons. A little bit different. Phospholipids. Talked about phospholipids just a minute ago for forming cell membranes. And if you look at them, they have a phosphate in the head and two lipid tails, one's saturated, one's unsaturated. By having the phosphate here, it's called a phospholipid. Look at the tails. Look at what the head is, hydrophilic head. So the phospholipid molecule has a hydrophilic head, and you know the lipid tails are hydrophobic. So one part of this molecule is hydrophilic, water-loving, and the other part hydrophobic, water-fearing. This little drawing over here on the right is how we draw it, so it's easier to draw just on a piece of paper. This is the hydrophilic head, and here's the two tails. Just draw two little lines down here for the tails. Now, I told you that these make up cell membranes, and you know that cells have cytoplasm on the inside. That's got water, so the hydrophilic head has to be directed toward that. Well, there's also capillaries leak fluids over the tissues, so the tissues are moist also. There's water out there. So how's this going to be built? Well, I told you it's a phospholipid bilayer. So let's look at the next slide. Here it is, phospholipid bilayer. The phospholipid heads are directed toward water outside the cell, and water in the other layer, the heads are directed toward the cytoplasm inside the cell. The tails are as far away as they can get. They're in the middle of the membrane. So this is initially how cell membranes are structured. They're a phospholipid bilayer. The phospholipid molecule has a hydrophilic head and a hydrophilic tail. The heads are directed toward water. The tails are as far away as they can get, the middle of the membrane. Here's some steroids, and you can see that there are 4 ring structures here that are identical. The top, they both have a hydroxyl. They both have a methyl. The only difference is that testosterone has a methyl group and a double-bonded oxygen on this end, and estrogen has a hydroxyl. That's it. That's the difference between estrogen, which brings out the female characteristics in this duct, and testosterone, which brings out the male characteristics in this duct, this male duct. Now, females have different traits than males. You know that, right? Females have, most of them, have less body hair. They don't have facial hair like guys do. They can't develop musculature like guys because guys have testosterone, and it allows them to develop a little bit differently. So the steroid has a big effect on the overall secondary sex characteristics of males and females. Proteins, composed of carbon, hydrogen, oxygen, nitrogen, and sulfur. The primary monomers, the building blocks, are called amino acids. So amino acids are the building blocks for proteins. There are 20 different amino acids. Now, you're not going to know all the names of all the amino acids, not in this class. When you get into micro and beyond, you will have to know some of them. Amino acids are linked into chains called polypeptides. Poly means many. Peptide is the bond that's forming between the two amino acids when they're joined together. It's a peptide bond. So many peptide bonds when you join amino acids together. Remember, I told you use poly for 3 or more, right? Because you can have dipeptide. That's just 2 amino acids put together. But a polypeptide is 3 or more. The function as structural components, as enzymes, facilitate, that means helps the movement and for food storage. Wow, so they have a lot of functions. You know, meat is protein, right? Well, this movement here, there are myofilaments and there's myotubules in there too, and they're both involved in movement inside of cells. Well, here I have an example of some amino acids here, and let's just look at the next slide for just a second. See how they're all put together by condensation? Every one of them, they're joined together by condensation. So let's look at the picture. Here's what I was telling you about the gene code, genetic code. There are 20 amino acids, and they're abbreviated in here. So this is a table showing all the codons, which are these 3 bases, for the 20 amino acids used to code for proteins. So they're just abbreviated, and it's just showing, you know, leucine, whatever, valine, alanine, threonine, protein, serine, tyrosine. So they're all in there, but we'll look at that toward the end of this class. So here's the previous image, and it's showing the 4 amino acids here. Here's the molecule that we're looking at for the amino acid, and we're going to see a little more information on the next page, but you can see that there are 4 of them. And they're going to be put together by condensation. So let's look at the next slide. All right, every amino acid, every one of these that we have here has a central carbon in the center, a carboxyl group, C double bond OOH, there it is, a hydrogen, there it is on the bottom, and side chains. Oh, I put my amino group. Amino's over here on the left. There's your amino NH2. There's your carboxyl, C double bond OOH. There's your hydrogen. And up here at the top, this is the side chain. That's what makes one amino acid different than the other, is that side chain. And so when they're put together, there's your hydroxyl taken off of one amino acid, off the carboxyl of that one, the hydrogen off of the amine of the other one, put together to make water, and those are going to be joined together. That'll be a disaccharide. We join that to a third one here by condensation, polysaccharide. Put the 4th one on, polysaccharide. Every time you join them together, a molecule of water is produced through condensation. And that bond is called a peptide bond. It's going to be forming there. It's called a peptide bond. Now, protein structures. Structure is critical to the function of an enzyme. Just like any of these molecules I've talked about, their structure is essential to their function. They're structured differently and they have different functions. And it's very important that it be shaped correctly. Just like keys, you can have 100 key blanks and they could all be cut differently with different notches in them. They're all different, but they're the same blank. They're all keys, but they have different notches in them, so they don't all work the same. So structure is very important. The primary structure, number, kind, and sequence of amino acids in the polypeptide. This is going to be a linear sequence of the amino acids. It's going to be like in a chain. So look at the next slide. Here it is, primary structure. Here's the amino end, and all these amino acids have been put together all the way down. Here's the carboxyl end, acid end. And when you put another amino acid on here, the amine's going to join to that carboxyl, and this carboxyl end will be over here. So one end is always going to have an amine on it, the other end is always going to have a carboxyl. So it says the primary structure of a protein is linear, with an amine group at one end and a carboxyl end at the other. So it's like a shoestring. You can use a shoestring is what I would bring to class. Secondary structure, the repeated pleating or coiling of the polypeptide. Well, the repeated pleating would be like a piece of paper that you fold on itself time and time again, and it makes a corrugated or a rippled appearance to it, like this right here, back and forth, back and forth, back and forth. That pleated sheet, that's one form. And then the helix is like a telephone cord. If you have an old telephone cord, it's kind of spirals. It's a double helix. It coils. So it goes round and round, okay, like a coil. So you have secondary structures that are alpha helixes and pleated sheets. Those are secondary structures of proteins. The tertiary structure, three-dimensional folding of the polypeptide. Okay, the tertiary structure is going to be a combination of primary and secondary structures. So it's going to be going all over the place. See, primary and secondaries together. Here's your tertiary structure right here. So it's a tertiary structure made of primary and secondary structures, and it has a particular shape. It's going to form into an exact shape that it needs to be formed in to perform the function that it was made for. So it's made specifically to do something, and these alpha and betas and pleated sheets all go together to help it to assume the shape that it's supposed to be. Now the quaternary structure, the linking of two or more polypeptides to form a complete protein. So quaternary, that's the fourth quad, quarter is a fourth. This is the fourth structure. The linking of two or more polypeptides, two or more tertiaries, to form a complete protein. Okay, so just because it's tertiary doesn't mean that it's going to be working by itself. It may have to be joined to another molecule to work. So let's look at that one. Here is collagen. It's made of 3 strands of a polypeptide chain make up the molecule called collagen. And collagen is what gives your skin its firmness, your bones this firmness. Collagen's in cartilage and tendons. So it's what gives your body the firmness while you're young. When you get older, your body makes less collagen. That's why your grandparents are kind of soft. They don't have as much collagen in their tissues anymore. And then here's hemoglobin over here. It's made of 4 molecules, 2 beta chains and 2 alpha chains, and they have to have iron in there to carry the oxygen. But this is a quaternary molecule of hemoglobin. It's made of 4 tertiaries put together. Here's just another imagery here showing the linear for the primary. This only shows the alpha helix for the secondary. It doesn't show the depleted sheet. Here's your tertiary. It's going to be a combination of alpha helix, that secondary structure, the depleted sheets, and the primary structures. And then your quaternary, this showing hemoglobin again, made of 4 of your tertiaries, 4 polypeptides put together. So we get to nucleic acids. Now see, they're composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And remember, proteins had sulfur. Nucleic acids have phosphorus. The primary building blocks are called nucleotides. So the primary building blocks, the monomers of nucleic acids, are called nucleotides. And they have 3 parts to them: a 5-carbon sugar, a nitrogen base, and a phosphate. Now, this nucleotide having those 3 parts is like your hand. When you hold up your hand, doesn't it have parts? It has, a thumb, a pointer, a flipper, a ring finger, and a little finger, and then a palm. Those all make up your hand. Well, a nucleotide has 3 parts. A 5-carbon sugar, a nitrogen base, and a phosphate make up a nucleotide. Now, deoxyribonucleic acid, which is abbreviated DNA, that's the genetic material in all cellular organisms. And it is a double-stranded molecule. It has 2 strands. RNA, which is ribonucleic acid, structural component of ribosomes, and this is ribosomal RNA, so it's nucleic acid for ribosomes. Involved in energy transfers, which we're not going to really talk about that much, respiration and photosynthesis. Involved in translation of genetic codes into proteins, we'll look at these. Messenger RNA, which is going to be a strand that's copied off of the DNA. Transfer RNA, which is going to be a special RNA that's going to bring in amino acids to build the polypeptide. And RNA is single-stranded. So we're going to look at several of these. These will be introduced mostly toward the end of the semester. Here's the nucleotide picture that I talked about just a minute ago. It has a 5-carbon sugar. There's deoxyribose. It has a phosphate and a nitrogen base. Well, nitrogen bases are almost the same in DNA and RNA, but there's one base difference between DNA and RNA, which we'll see in just a minute. Here are the 2 sugars, deoxyribose and ribose. Can you tell a difference between those? If you look kind of close, you can find out where the difference is. Look at ribose. Carbon number 1. Carbon number 2 has a hydroxyl on it. But on deoxyribose, which means without an oxygen, it's missing one, carbon number 2, it just has a hydrogen, so it's missing the oxygen. This is why they call it deoxyribose. It's missing an oxygen. That's the only difference between those 2 sugars. And one is used to store hereditary information, and the other one is used for making proteins. Now the bases. Remember, a nucleotide has a 5-carbon sugar, a phosphate and a base, a nitrogen base. The base pair sequence, there's always a purine and a pyrimidine. I see the pyrimidines are over here, the purines are over here. So it's 1 to 1. It's always a purine and a pyrimidine that are going to bind with each other, base pair. Now in DNA, adenine pairs with thymine. Adenine pairs with thymine. In DNA and RNA, both of them, guanine and cytosine base pair. Cytosine, guanine. So they both have guanine and cytosine. In RNA, adenine pairs with uracil. Now uracil is not shown here. They never do for some reason. But it takes the place of thymine. It's a pyrimidine. Okay? So there's one base difference. In DNA, adenine pairs with thymine, whereas RNA, adenine pairs with uracil. So one base difference between the two. Now I kept those straight as far as knowing who are purines and pyrimidines. When I told you about mnemonics you can do, well, when I was taking cell, I had to remember this stuff, but that's why you start getting frantic about mnemonics. And I had to remember which ones were single rings and which ones were double rings. And it hit me I could use what's called opposite day. It's opposite of what it is. The short word, purines, compared to pyrimidines, the smaller word was the bigger molecule, had 2 rings. And the long word was the little one, had 1 ring. So it was opposites. The longest word was the single ring. The shortest word was the double ring. Okay, that one hit pretty good. And then know which ones are purines and pyrimidines. Well, if you look at the word pyrimidine, you see a Y, which just happens to also be in cytosine and thymine. And you know that uracil replaces thymine, so it's going to be there too. So you could tell which ones were pyrimidines easy just by knowing how they had the Ys versus the purines, which were the other ones. Here is just an example of a nucleotide chain. This is one chain of a double chain of DNA. So the other chain would be over here on this side, over here. So it says nucleotide chain. Nucleotides are joined together to form nucleic acids. All right, here is one nucleotide. Here's a 5-carbon sugar, a nitrogen base, and a phosphate. Here's the second one, 5-carbon sugar, nitrogen base, and a phosphate. A third one, a fourth one, a fifth one, and a sixth one. So there are 6 nucleotides there making up that chain. So it says nucleotides are composed of a 5-carbon sugar, the blue, a nitrogen base, the purple ring, and a phosphate, the yellow. So it points them all out there. That's one side. Now, there's the next picture is going to show that there's a ribbon that replaces the sugar phosphates because they're all the same. If it's DNA, that's deoxyribose sugar phosphate, deoxyribose sugar phosphate. What's the difference is the code is in bases. So look at the next picture. Okay, on this one, you can see there's a ribbon here which represents the alternating sugar phosphates. What you see in between are the nitrogen bases, and they have hydrogen bonds in between them. They're holding those together. And hydrogen bonds are weak electrostatic attractions, so they can be taken apart pretty easy when the DNA needs to copy itself or a gene needs to be exposed. So hydrogen bonds between the nitrogen bases provide the force needed to hold the 2 strands together.